Good afternoon, everybody. Welcome to the Essex Library Zoom event with What You Should Know About Ticks and Tick-Borne Diseases with Dr. Kirby Stafford. My name is Ann Thompson. I'm the Assistant Director for Public Services here at the Essex Library, and I'm very, very delighted to welcome you all to our meeting today with Dr. Stafford. Just a few housekeeping notes to kick off with. Please keep your cameras off and your microphones muted just to save bandwidth so that we can keep a very clean recording, which we'll post to our YouTube channel afterwards. Please type your questions as you think of them into the chat, and we'll get to them after Dr. Stafford's talk. We can also have audio questions afterwards too. We'll get to them all. I'm going to launch a poll so that I can see. I'm asking how many people are viewing your screen with you today so we can get some more accurate numbers for our attendance to give to the State Library next year. And I would be very pleased and honored to introduce you to Dr. Kirby Stafford. He's the Chief Scientist of the Department of Entomology and State Entomologist at the Connecticut Agricultural Experiment Station. Research area is the ecology and control of the black-legged tick, Ixodes scapularis, if I pronounced that correctly, and more recently, the Lone Star tick, Amyoma Americanum. Dr. Stafford has authored or co-authored 86 articles in peer-reviewed scientific journals, peer-reviewed chapters on tick management in 2 books, produced a tick management handbook, and serves on regional and national tick-related committees. Dr. Stafford obtained his PhD in medical veterinary entomology from Texas A&M University and a Master of Science degree in entomology at Kansas State University and a Bachelor of Science degree in entomology at Colorado State University. And with that, welcome, Dr. Stafford. And I'm going
Thank
to
you.
Poll and get that off the screen. And we'll kick it off.
Is it still up there?
It's gone. I think you may have to click close it out.
There it is. Okay. All right, welcome everyone. Good afternoon. I do have a lot of material I'd like to cover this afternoon, covering everything from about ticks as well as highlighting many of the diseases themselves. So let's get started. So this is kind of an introduction: tick-borne disease, morbidity, and mortality. And each year in the US, there's over 500,000 human cases of tick-borne diseases. And there are some fatalities caused by a few of these, primarily Rocky Mountain spotted fever, the Powassan virus encephalitis, and a handful of cases of Lyme disease, carditis, as you can see, in the various headlines that are shown here. The number of reported cases of Lyme disease to the CDC in the United States, just going back to 1998 here up through 2019, has been steadily increasing in a somewhat stepwise pattern. And I expect these numbers will probably continue to increase. And but it's not just Lyme disease. Ixodes scapularis, which is our black-legged tick, or as many of you know it as the deer tick, is an increasing public health concern because it's not just Lyme disease. I mean, if you look at the case of discovery of the pathogens associated with this tick, you have babesiosis, Lyme disease, anaplasmosis, the Powassan virus that causes a viral encephalitis, a new tick-borne relapsing fever Borrelia, and it just kind of goes on. So why are we dealing with this huge, steadily increasing number of tick-borne diseases associated with this tick? And the history really goes back quite a ways. And what I want to introduce here is the Travels of Pierre Combe. This was published in 1770. He came through in the mid-1700s. And in his journal, he noticed how bad the ticks were. To which these I must add the wood lice, that is ticks, which the forests are so pestered that it's impossible to pass through a bush or sit down, though the place be ever so pleasant, without having a whole swarm of them on your clothes. Interestingly, a century later, the state entomologist in New York, Asa Finch, noted that the most common tick of our country, the wood tick, though formerly abundant throughout the northern and middle states, has now become nearly or quite extinct. At this day, along the route that Combe pursued, not one can probably be found. So why? What happened during that intervening century. Well, if you look at Connecticut's forest history on the percent cover, you can see through the 16 and 1700s, there was a steady decline in our forest cover. Agriculture reached its peak around the 1830s or so, and then as lands opened up out west, followed by our industrial revolution, farms were abandoned and the forest through the 1900s steadily came back. Today, Connecticut is about 58 to 60% forested. So basically we had habitat, very little habitat, and then of course the habitat came back. Along with that was our population of white-tailed deer. It's estimated in 1896 there was only 12 deer in Connecticut. Today, the estimate is probably somewhere well over 100,000 animals here in Connecticut alone, based on surveys or estimates by Connecticut DEEP. So basically, we now have the habitat and we have the host. So in the mid 1970s, then we saw emergence of this new disease. So the recognition of Lyme disease in Lyme and Old Lyme, Connecticut, re-triggered a focus on human tick-borne diseases. But interestingly, this disease had a long history in Europe. The first recorded Lyme rash with a tick bite, although that connection wasn't really made, was in Europe, was a paper out of Sweden in 1909. And basically, a few women, particularly Polly Murray, who moved to Lyme, Connecticut in the late 1950s, all through the 1960s and 70s, she noted that her husband, 4 children, and herself were periodically plagued with mysterious symptoms. In time, I came to suspect that these ailments were somehow linked. And she actually published an interesting book on those early years called The Widening Circle. A Lyme disease pioneer tells her story. That was published back in 1996. But the story actually goes way back further than that. So I often ask people, do you know what the oldest known human Lyme disease case is? It's actually Otzi. Okay, as you can see here, the most ancient person ever diagnosed with Lyme disease. He was the Iceman, who basically, so this is a case dating from at least, 5,000 years ago. So as we look at the expansion of Lyme disease, probably what we think happened is the tick itself survived on the eastern end of Long Island, islands off the Cape where deer were not hunted out. And then as conditions returned, as I alluded to earlier, the tick began to expand its range. And of course, the Lyme area is one of those early areas. So this map here shows you the distribution of Lyme disease cases in Connecticut based on laboratory testing here at the Connecticut Agricultural Experiment Station, working with the Department of Public Health. And even at that time, you can see the greatest incidence of cases there was right there east of the Connecticut River. And as you can see in these subsequent maps, then you can see how Lyme disease cases expanded here by 1996 and all through the Northeast and upper Midwest here by even, 2018. So we've seen a steady geographic expansion of the tick and of course Lyme disease along with it. So today we estimate only probably about 10% of the diagnosed Lyme disease cases actually end up getting reported and documented. So the true incidence of Lyme disease is probably somewhere around 476,000 to 500,000 cases. But what I want to point out is that this part of the pie of vector-borne diseases with other tick-borne diseases, which currently is around 26, 27 or so percent of the total tick-borne diseases, is steadily increasing. And if you look at this over time, I want you to just look at the trend. Don't try to concentrate on the specific diseases here. But the point is, from the recognition of Rocky Mountain spotted fever in the late to early 1900s, which was the first human associated tick-borne disease recognition there, you can see there's been a steady history of discovery of additional tick-borne pathogens that cause human disease, with an acceleration in cases in most recent years. The blue here are those that are carried by our Lone Star tick, green by our black-legged tick, and of course in brown are the ones carried by the Dermacenter species ticks, the dog ticks, or Rocky Mountain wood ticks. So there's been a steady increase in the number of discovered tick-associated diseases in humans or pathogens. And so here we've got, we're approaching around 20 known tick-associated diseases. I'm not going over all of these specifically. I'm going to concentrate on a few of concern here in the Northeast shortly, but nonetheless, you can see there's quite a handful of tick-associated diseases carried by these three genera of ticks here in the United States. So here in the northeast or eastern United States, of course, our main tick of concern continues to be the black-legged tick, Ixodes scapularis, which is the main vector for Lyme disease. We have our common American dog tick, Dermacenter variabilis, and this has actually been a very active year for this particular tick. And then we're also seeing some native tick species expand their range, which is exposing more people to these ticks. And so this would include the Lone Star tick, the Gulf Toast tick, and then we also have the new exotic invasive tick species, the Asian longhorn tick, Hemophilus longicornis. So to highlight these and go over these briefly, I just want to point out for those of you that perhaps subscribe to Connecticut Wildlife back in the September-October 2020 issue, I did have a brief article on the emerging and exotic ticks here in Connecticut, particularly focusing on the Lone Star tick and Asian longhorn tick. So let's just briefly go into these other ticks that we're starting to deal with here. The Lone Star tick, Amyoma Americanum, is responsible for 90 to 95% of all the tick bites in the southeastern United States. And it has its own suite of diseases or pathogens that are associated with it. The primary one is ehrlichiosis. We have several viruses, as well as red meat allergy or alpha-gal syndrome. And I'll go back into these here just in a moment. But we're seeing a steady expansion of this tick northward into the Northeast and Midwest. Now, interestingly, we just never picked up this tick in Connecticut. However, we were starting to see a slow but significant increase of submissions to the tick testing laboratory here at the experiment station, mainly from Fairfield County. Then in June of 2017, we had a report of a dead or dying deer on Manresa Island in South Norwalk. And so these were a picture that was taken by an environmental conservation officer from DEEP that was sent to me. I joined up with him the next morning and confirmed that all of these ticks on this poor animal were Lone Star ticks. This was a heavy, extremely dense population of this tick right at that particular locality. And we've been seeing a steady increase, even though small, of those ticks coming in to the United States. Now, the part of this has to do with our warming winters. So if you look at this map here, where the NOAA data was used by the Washington Post to show where that every Northeast state except Pennsylvania, the temperatures in the winter months had risen by 2 degrees Celsius since 1895, 1896. And interestingly, the Lone Star tick actually first moved into Long Island, and starting in the early 1990s, and is extremely abundant there. It's starting now to show up on some parts of the Cape Cod and islands off the Cape. And again, like I said, we found it here in Connecticut. And like I said, this has to do with overwinter survival, we think. Here in Connecticut, we found the average overwinter survival of adult Lone Star ticks was 63%. And interestingly, even up in coastal Maine in the Portland area, and some overwintering studies that we did, when you had leaf litter or snow cover as insulators, 28% of the ticks survived even up in Maine, even though the tick is not established there yet. So actually, we published a paper in the New England Journal of Medicine, just noting here where we're starting to see expansion of this tick here into New England. The other tick we're keeping an eye on, of course, is the Asian longhorn tick, Hemophilus longicornis. This is an East Asian tick. It was discovered on a sheep named Hannah at a farm in Hunterdon County, New Jersey, back in 2017. This tick is considered a serious pest to livestock, including cattle, horses, sheep, and goats, and will attack pets like dogs, wildlife, and very occasionally humans. However, it became established in Australia and New Zealand by the early 1900s, where it's a major cattle pest, but it is also known as a vector for several human and animal disease pathogens, and its natives range parts of China, Korea, and Japan. The interesting thing about this tick is it reproduces parthenogenetically. No males required. So it can consequently build up a huge population relatively quickly. So since that first discovery, it's been reported now in 15 US states. Actually, I think that's up to 16 now since I made this slide. We have a couple of pies, very abundant in New Jersey, Staten Island, parts of Westchester County, New York. We have found a couple of very heavy populations in Fairfield County and have picked up individual ticks, larvae and nymphs, through all of the southern Connecticut counties. It is not a vector for Borrelia burgdorferi, the spirochete that causes Lyme disease, but it can transmit the agent for Rocky Mountain spotted fever, at least in the laboratory. At this point, it's really mainly a veterinary concern. It doesn't really tend to, it will bite humans, but doesn't seem to readily prefer or like humans. So it's definitely something we're keeping our eyes on. Another tick that we're keeping our eyes on is Amyoma immaculatum, the Gulf Coast tick. Its original distribution really literally was along the lower Atlantic and Gulf Coast states, and it was primarily considered a livestock concern. But it is the main vector for Rickettsia parkeri, which is a milder form of spotted fever in humans, and also a hepazoa and Americanum, which causes a disease in dogs. And so its range continues to expand. And then back in 2020, a population was discovered along coastal Fairfield County. So it's another tick we're keeping an eye on. So how are we going to address these tick-borne diseases? And I like this quote from Dr. Dan Sonnenstein's textbooks on the biology of ticks, his second volume. And he notes that few agricultural health problems confronting human societies have proved as intractable as the control of ticks and the many diseases that they transmit. And I think this will become apparent as we kind of go through this. So what are the tools for tick control or tick management? Well, you have education and behavior change. Okay, that's kind of like part of what we're doing here today. Personal protection measures continue to be extremely important. We have extensively relied on chemical control, whether we're talking synthetic insecticides or there's a lot of current interest in botanicals or so-called natural compounds, which I'll just point out aren't that effective for the most part, but I'll get into that in a moment. There's landscape modifications to make the habitat a little less suitable for ticks. Biological control is an area that's been looked at. And there's been a lot of research on host reduction or exclusion, host-targeted acaricides, that is treating the mice and deer to kill the ticks that are on them, and even host-targeted vaccines. So like a Lyme disease vaccine for the mice. And I've done a lot of work with that, but I won't be talking too much about that today. Most of these tick control methodologies, of course, have been researched from a standpoint to control black-legged ticks because of the interest in Lyme disease, and have not really been found effective or evaluated in controlling these new ticks that we're dealing with, like Lone Star ticks. Now, all of these ticks that we're talking about are what we call three-host ticks. So there's three active stages of the tick. You have their larvae, your nymphs, and your adults. And each stage requires a blood meal before going to the next stage. So this is the life cycle of our black-legged tick, Ixodes scapularis, larva, nymph, and adult female here. And this is an engorged female laying her egg mass. So each engorged female tick can lay about 2,000 eggs. The larval ticks will hatch from that egg mass and feed primarily on rodents and birds, although they will feed on any animal. And they're not infected, say, for example, with the pathogen that causes Lyme disease, because it's not passed from the female tick to her progeny. They become infected if they feed on an infected mouse, chipmunk, a few species of birds or shrews. And after they feed, they drop off molt to the next stage, the nymphal ticks. The nymphal ticks, again, feed primarily on rodents and birds, but they will feed on any animal at all, including us. And then once they engorge, they drop off and become adults, male or female ticks, which feed only on larger animal hosts. So we're talking things like dogs, cats, us, foxes, raccoons, things like that. But the primary preferred host is white-tailed deer. And again, once that engorged female drops off, she lays 2,000 eggs, completing the life cycle. So why is summer the highest risk? So you just saw those various stages of the tick in the life cycle. Okay. Now the part of it, principally is because of your contact risk, because obviously more people are active in the summer versus your spring or fall or winter time. In the fall, as temperatures drop, you'll be wearing more clothing. And you also have, given that population, you have higher numbers of nymphs versus the adult female ticks that you'll encounter in the fall. And that's because they're much smaller in size. So this is an engorged nymph showing it with a straight pin for size comparison. They're a little bit cryptic because they are so small. The adult, here's an attached adult female, a little larger, easier to spot. And transmission by an infected black-legged tick begins after about 24 to 36 hours of attachment. So about 69% of the adult females, but only 47% of the nymphs are removed within that first window of transmission because they're easier to see. And why is summer the high period of risk? This graph right here shows you the seasonal activity of the ticks. So the eggs are laid by the female ticks generally around May, even if they fed in the fall or in the spring. Those larval ticks will hatch from those eggs around mid-July. So August is your peak month for larval tick activity. Those larval ticks feed, and again, they drop off, molt to nymphs, and then will go through the winter and show up the following year as nymphs. So the nymphal ticks you were facing this summer were larval ticks last August. Once those nymphal ticks feed that are active here with May, June, and a little bit into August being the peak period, they drop off. Those nymphs then become adults that show up here in the fall. The adult stage does not hibernate. It will can be active in the warmer days through the winter, and then you get a smaller peak in the spring. As things warm up, those adult black-legged ticks will become active again. These are the ones that have not yet found a host. Now, if you look at human case data from CDC, you can see that the peak number of Lyme disease cases occurs here in the summer months of May, June, and July, exactly when the nymphal tick is active. Far fewer cases are associated with the adult stage of the tick. So that gives you kind of a seasonal feel for what your risk lies. But what about your risk of actually encountering ticks in the landscape? Based on surveys that have been done both in New Jersey and here in Connecticut, these surveys asked, people where were they when they picked up the tick that was submitted to the experiment station for testing. 74% said they were outdoors right at home. 20% were involved in activities away from the home. And somewhat similar statistics were found in New Jersey as well. If you ask people what they think they were doing when they picked up that tick, you'll notice play outdoors accounted for nearly half. So if you look at the Lyme disease statistics by age class, you'll notice that children actually have the highest rate of Lyme disease. And so other high risk activities obviously would be things like gardening and yard work, and then for a less extent, hiking. Any outdoor activities can put you at risk of running into a tick. So basically, some places you'll see these alerts, tick alert. Ticks carrying Lyme disease can be found in this area. Examine skin, clothes, and pets thoroughly. If you look at Lyme disease cases here in Connecticut, you'll notice that there's a big increase. This was a drop due to change in reporting, which then was resumed with here in 2007 and 8. But you can see over the past quite a few years, we've been seeing a steady decline in the number of reported Lyme disease cases here in Connecticut. But you're still talking a little under 2,000 cases reported. But that still translates to roughly around 20,000 or so cases each year in Connecticut. It's very unclear why we're seeing that decline. I like to think maybe people are being more conscious about taking tick bite prevention measures, but it could also be an issue in terms of fewer physicians actually reporting the cases they're diagnosed, but we really don't know why. So we actually do two types of tick surveillance. We have what we call passive surveillance, which are those ticks that you all submit for identification or testing. And then active tick surveillance, we're actually doing the tick sampling. And the standard method of collecting ticks is dragging the vegetation with about a square yard of a white flannel-like material. The ticks will grab a hold of it just like they would you or your pet. And it's an easy way for us to track the ticks. And then what's really kind of interesting is that Popular Science back in their November of 2004 issue said that tick collecting was one of the worst jobs in science. Well, I didn't think so, but in any event, here's a picture of me testing, looking at the tick drag for the small ticks. And then for some tick species like Lone Star ticks, you can use a carbon dioxide trap because they are attracted by CO2. Doesn't work well for our black-legged tick, but it can work for Lone Star ticks. And then you can also live trap the animal hosts and count the ticks that are on them and then re-release them, which is what we do with the mice. So when we're talking about these host-seeking ticks, bear in mind that 90% of their life cycle is spent on the ground. They're either looking for a host, they're digesting that blood meal they just got, they're going through the winter, and so they're spending most of their time down there in the environment. Bear in mind, ticks do not jump, they don't fly, and they're not dropping from the trees. They actually assume a questing position. For the nymphs and larvae, they're down in that leaf litter or the grass tips like you see here. And then they'll hold out their front pair of legs ready to grasp when you walk by. So the immature ticks, the larvae and nymphs will be in the low vegetation because they're feeding, mainly on ground foraging birds and mice and chipmunks. And the adult ticks can get into that shrub layer, because remember, white-tailed deer is their preferred host. And they have a variety of sensory receptors that can detect the host. They have chemical cues like the carbon dioxide, ammonia, lactic acid that you're emitting. They can detect body heat. They can detect the vibrations as you're walking along. And there are some ticks they can't really see, but they can pick up visual cues like shadow through their light sensing cells. So as we look at the passive surveillance results, these are the ticks that were submitted to the tick testing laboratory here at the Connecticut Agricultural Experiment Station, which is overseen by Dr. Gudars-Mallet. And generally, you can see that the number of ticks submitted have ranged from a little over 3,000 to almost 6,000 ticks each year. And these are the results through 2019. I haven't added the 2020 numbers here yet. But in any event, what I want to point out is that, roughly around half, you will see, of those ticks are not infected. And roughly around 30% overall are infected with Borrelia burgdorferi, the bacteria that causes Lyme disease. A little around, just a little under 10% are infected with Babesia microti, which causes babesiosis. And somewhere slightly lower rate, between roughly 5 to 12%, are infected with the agent Anaplasma Phagocytophylum. And then you get lower rates of co-infection. So you can see here that 5% of the ticks were carrying Borrelia and Babesia, which means that particular tick could infect you with 2 disease pathogens at once. And then you got other mixed combinations of co-infection. And then in very low cases, you'll actually have the rare tick that has all 3 pathogens present. If you get bit by that tick, you just unfortunately won the tick lottery here. So conversely to passive surveillance, where we just wait for you to submit ticks, we have an active tick surveillance program that started in 2019. And we continued this in 2020 and 2021. It's funded from funds from the CDC that come through the Department of Public Health. We have 40 sample sites around the state and all distributed through all 8 counties. In 2019, we collected a little over 2,000 black-legged ticks, in addition to American dog ticks, just a few Lone Star ticks, and 2 Asian Longhorn ticks. In 2020, we collected a total of 2,000 black-legged ticks, again, a number of American dog ticks, a few more Lone Star ticks, and a few more Asian Longhorn ticks, and all of these ticks are tested here at the Agricultural Experiment Station. I haven't put in the 2021 collection data yet at this point, but it's very similar to 2020. If you look at the infection rate in the Ixodes scapularis adult females and nymphs, and here I've actually broken it out as just rather than a lump sum infection rate, you can see for Borrelia burgdorferi, the Lyme disease bacterium, you can see that 48% in 2020 of the adults were infected, only 20% of the nymphs were infected. And similarly, you always see a slightly higher infection rate generally here in the adults versus the nymphs, because you have 2 opportunities to become infected, once when the tick feeds as a larva, and then again feeding as a nymph. So the point is, not every tick that you pick up, again, is necessarily infected. And then, of course, the rate of infection by the Powassan virus is very, very low on average. So your first line protection is your personal protection measures. If you're doing high-risk activities, even though it's hot, wear long pants, tuck them in the socks. Remember what I said about where the ticks will generally be picked up on the lower extremities, and you'd be surprised how fast they can climb up. Use a repellent. Bathe and check for ticks when you come back indoors. And those tick checks are really critical, and then promptly remove any ticks. As far as the repellent goes, you can use DEET or picaridin or oil of lemon eucalyptus for skin-based repellents. For clothing, you can use these as well, but permethrin is available as a spray, 0.5%, and you can also buy from several companies clothing that has been pre-treated with permethrin. Permethrin is an insecticide. It acts as both a repellent and toxicant, and any ticks that come in contact with your clothes are killed. It can be very, very effective. It's what I've used over the years when I've been doing field work, collecting ticks, and I've had very few tick bites, and knock on wood, I have not yet gotten Lyme disease. So that is certainly a very, very effective approach when you're engaging in yard work or other high-risk activities that would expose you to ticks. Now let's talk about the tick itself. These pictures show you the mouth parts of the tick. So this is an Ixodes scapularis nymph, and this is an adult. And if you look at it here, the mouth parts here are 3-pronged in structure. So basically you have what we call two cheliceraal digits. And you can see the teeth on the end of these. This is what the tick uses to cut into the skin. And then you have a hypostome, which is kind of like a lower lip with all these recurved denticles on it. Again, if you look here at the scanning electron micrograph of the head of a adult tick here's the palps on the head of the tick. They're like antennae on an insect. They're sensory in function. Here are those 2 chelicerae. Here is the oral cavity that the tick feeds through. And again, here's that hypostome, which has all these recurved denticles on it. And here is just a lower shot of the mouth parts of a Lone Star tick, Amyoma Americanum. And here you can see very well all those recurved denticles on the lower side of the hypostome. So when a tick feeds, those Lyme disease spirochetes are in the tick gut, shown here in green. And they actually have to migrate through the body of the tick to the tick's salivary glands before they get injected in the feeding process. So when a tick isn't like a mosquito, with hypodermic mouth parts. You know, a mosquito feeds for what, 1.5, 2 minutes, and it's gone. A tick feeds for day. For like a nymphal black-legged tick, when it attaches, it requires 4 days for full engorgement before it will drop off. A female black-legged tick requires 5 to 7 days for full engorgement before it drops off. And so a tick has to set its mouth parts. So once it cuts into the skin and does that, it secretes a cement-like material. Some ticks secrete a lot of the cement. Black-legged ticks only a little bit. But in essence, it's sort of gluing itself to you as it embeds those mouth parts. And so then it begins salivating, and it secretes a lot of pharmacologically active materials, proteases, anticoagulants, vasodilators. It creates this little microscopic feeding cavity or lesion right here under the skin. As the blood pools in there, the tick is spitting and sucking through the feeding process. Now, again, that takes time for those spirochetes to reach the salivary glands and get injected in. In addition, they change the outer surface proteins they express from outer surface protein A to outer surface protein C. So there's actually a change that takes place that's required for infection. So about half the ticks for most people, based on surveys, are removed in the first 24 hours. So basically at 24 hours, your risk of transmission is basically 0%. Rare cases with a lot of ticks, it could be up to 5% transmission rate, but it's generally 0. By 48 hours, you're looking at 11 to, in rare cases, up to 68% transmission rate. And the longer the tick is attached, you can see a much greater rate of transmission from an infected tick. And this is, of course, for Borrelia burgdorferi and an estimated duration of attachment for a nymph parasitizing humans. But I would say, by contrast, the Powassan virus has been shown to occur in 63 to 75% of the cases within 15 to 30 minutes. So that's one of the rare cases of rapid transmission. And the other pathogens like Babesia and Anaplasma still take roughly around that 24-hour period for transmission. So the take-home message here is regular tick checks and prompt tick removal. That's one of the take-home message. The risk of spirochete transmission increases with the length of time the infected tick is allowed to remain attached. So I like to use fine-tip forceps and grasp the tick as close to the skin as possible and gently and firmly pull it out. Save the tick, and you can submit it to the station for tick testing. Now, what if the tick's infected and you don't get it back in time? Then you get Lyme disease. So basically, about 70% of the people or so will develop the characteristic Lyme rash or erythema migrans rash. It's a slowly expanding red rash. It appears generally 2 to roughly 7 days after the tick bite, although it could be as long as a month. And occasionally it will clear in the center. The actual bullseye form only occurs in about 15% of the cases. It has very little sensation, although it could be warm to the touch. So if it's on a part of the body you're not examining, you could actually miss it. And then if you're not treated right away, some people will develop facial palsy, like you see here. That's paralysis of your facial muscles. 1% carditis. You can develop neurologic Lyme, meningitis or encephalitis, or radial neuropathy, which is tingling in your fingers and extremities. And then, of course, about 28% or more will develop the characteristic Lyme arthritis, primarily in your larger joints like the knees. And about 20% of people that are not treated very early with antibiotics can develop these ongoing unresolved symptoms. So babesiosis, we have babesiosis and anaplasmosis. Now, interesting, babesiosis is caused by Babesia microti, and this is actually a protozoan which infects your red blood cells. It's sort of like having malaria. Its main symptoms are fever, chills, sweats, aches, nausea, and fatigue. It can range from very mild to very severe. And about a quarter of adults and half of children will be totally asymptomatic. You won't even know you have it. But it can be severe or fatal in elderly or immune suppressed individuals or people that do not have a spleen. And it's treated with a different set of drugs. Anaplasma Phagocytophylum is a bacterium. It infects granulocytes, which is a type of white blood cell. And you can see a picture of an infected white blood cell here. Again, fever, fatigue, muscle pain, chills and sweats, headaches are typical symptoms. It can be mild or extremely severe, particularly in older individuals. There's no rash or anything like that. And what may be a tip-off is that in your lab work, you'll see a decreased white blood cell count or platelet count. And it's readily treated with doxycycline or tetracycline as long as you get a proper diagnosis, but it can be quite severe in some individuals. The Powassan virus is kind of interesting. This virus was first described in 1958 in Powassan, Ontario. It's closely related to the West Nile virus, and there's actually what we consider two lineages. The classical lineage 1 strain was transmitted by Ixodes cookii, which is the groundhog tick or squirrel, and Ixodes marxi, the squirrel tick. This tick occurs on groundhogs and skunks and things like that. Occasionally does bite humans. And then the deer tick virus was discovered, which is transmitted by our deer tick, the black-legged tick, Ixodes scapularis. And with that, we're starting to see, it's still relatively rare, but we are starting to see a number of increasing cases of neuroinvasive disease caused by this particular virus. Again, primarily in the Northeast, as you see here, and up in the upper Midwest as well, kind of overlapping with our peak Lyme disease areas. And if you look at cases, there were 8 cases in Connecticut from 2010 to 2019, 31 cases in Massachusetts, 20 in New York, 12 cases in New Jersey, and 68 cases up in Wisconsin and Minnesota. The 5 cases in 2019 in Connecticut were in Ridgefield, New Canaan, New Preston, and Newtown. There was 1 fatality. There have been 2 cases that I'm aware of that have been diagnosed so far here in 2021. It again is relatively rare. It has about a 10% fatality rate. So it's not to be taken lightly. Most people that are infected are probably like West Nile are asymptomatic, but those that do develop the neurologic manifestations, about half will have permanent neurologic damage or disabilities as a result of the disease. Switching to the Lone Star tick, as I mentioned, ehrlichiosis is the main disease associated with this tick. It's very similar to anaplasmosis. It infects a different type of white blood cell here, as you see, and the symptoms are very similar. Again, nondescript fever, fatigue, muscle pain, chills and sweats, and headache. Again, maybe mild or severe, particularly in older individuals. And again, you get that lower white blood cell count and platelet counts. And again, it's readily treated with doxycycline if it's properly diagnosed. But the one that gets a lot of interest is what we call the alpha-gal syndrome or red meat allergy. This is not a pathogen. This is an allergy to a sugar called alpha-gal. And this is a molecule that's found in most mammals, but not primates. We don't have this sugar. And so there's evidence that this alpha-gal sugar is found in the saliva of certain types of ticks, and it's been linked with the Lone Star tick. And this allergy can lead to a serious, potentially life-threatening allergic reaction, which occurs 3 to 6 to 8 hours after people eat red meat or exposed to other products containing alpha-gal. Symptoms can range from rash and hives, nausea or vomiting, to a drop in blood pressure, dizziness, faintness, severe stomach pain, or full anaphylactic shock. And the only way to avoid the red meat allergy once you develop it is to, of course, avoid meat, mammalian meat, and some dairy and mammal derived products like certain gelatin and things like that. You can still eat fish and you can still eat, chicken, for example, and it's not clear. It does seem to fade over time, but it's new enough that there's still a lot that we need to learn about this. Now, I've been talking about personal protection measures. Let's talk about actual methods of actually trying to kill or control ticks. And the primary one used today still is the application of acaricides. That is pesticides that kill ticks. And these can be synthetic ones like carbamates, like 7, pyrethroids. Some neonics are in formulations used for companion animals. There was a microbial biopesticide, an insect fungus called Metarhizium anasophyllae, available for a number of years, but it's currently not available. And then there's been a lot of interest lately in the botanicals, particularly products containing plant extracts or essential oils that are derived from the EPA's 25B list of minimum risk pesticides. Challenges for using these. I tell you, there's a lot of uncertainty about the use of these pesticides in the backyard. There's obviously some homeowners are very afraid or adverse to using pesticides for some justifiable reasons. There's a lot of lack of knowledge about acaricides and how to read a label by homeowners. There's a lot of spread of misinformation via the internet and personal sources about these materials. There's an issue of the use of effective materials versus ineffective products, and I'll get into these. That means basically EPA registered insecticides or acaricides versus these minimum risk materials. Of course, like any herbicide or insecticide type product, they can be misused or overused, and they do pose, some of them pose a high risk to pollinators. And of course, when you're spraying to try to control ticks, you're really limited in the scale or coverage that you can get that way. And interestingly, despite being able to prove that some of these materials kill ticks, we haven't been able to prove that they impact tick bites or actual the incidence of tick-borne disease. So this is a summary chart showing the % reduction of Ixodiscapularis nymphs by the application of acaricides to the environment. And as you can see, your pyrethroids here and your carbamate, carbaryl, or 7 is the common trade name for that. You can see that the reduction of ticks or control, it really can be quite high, ranging up to almost virtually 100%, close to or nearly 100%. The 25B products are less effective. There were a couple, like IC2 is a product, unfortunately, that's not available anymore, that did provide actually high levels of control. But as we move forward here, when we start looking at some of those EPA minimum risk pesticides, understand that the ingredients that are on this list by the EPA, and you can go to their website to look these up, include things like cedarwood oil, cinnamon, citronella, geranol, garlic oil, rosemary oil, and thyme oil, things like that. There's also a list of materials that are suitable for inert ingredients. And so a lot of new products are coming along that are formulated with these as active ingredients. And some trials that were conducted by my colleagues at the University of Rhode Island and published here in 2020, they found, here's your Talstar, which is bifenthrin. It's a pyrethroid, was used as your standard. So you can see you got 100% knockdown of the ticks and 100% residual activity in killing ticks with that. A few other products did relatively well. The original IC2 was also had a pretty decent rate of knockdown, 86%. A natural pyrethrum product with thyme oil and 2-phenylethyl propionate. Again, really good knockdown and good residual control. But what I wanted to point out here is they did test a couple of red cedar oil products, and it didn't fare too well. You're talking 0 to 5, about 5%, 5-6% knockdown, and very little residual activity as well. They had 83% knockdown with Nucatone, 35% residual. Nucatone, and I'll get to that in here in just a moment, but one of the things I want to point out before I do is you got to think about their impact on pollinators whenever you're making any kind of chemical applications. Bees, including the honeybees and bumblebees and our native solitary bees, are of course extremely important pollinators. Most of the current chemicals used for tick control are the pyrethroid insecticides, and please bear in mind, They're very effective in killing ticks, but they're also extremely toxic to bees that are exposed directly during application or to residues on blooming crops. So you got to be kind of careful how you use them. Even some of the botanical insecticides, often touted as being environmentally friendly, have been found to have toxic and sublethal effects to honeybees. So it's really important to assess these non-target effects even with those compounds. Now, getting back to the nucotone, nucotone is a component of the essential oil from Alaska yellow cedar, and it can be obtained from grapefruit and other sources as well. And I've done extensive work with this material. It's a very effective repellent and toxicant against black-legged ticks. It has now recently been registered by the EPA as an approved ingredient to be used to develop new insect repellents and insecticides. And so basically that means that this material is available for companies to develop their own brand name consumer products. And hopefully we will see some repellents and tick control products commercially available by, I hope, even next year. But this would be a very, very attractive option for controlling or repelling ticks with its tremendous safety profile, and it really does work. Now, another approach to trying to reduce your risk to ticks is landscape management. Remember, I talked about ticks are mainly down in that ground for most of their life cycle, so those ground conditions are critical to survival. Ticks require high humidity and that cover for their survival. So when you look at your residential landscape, you remember the Ixodes scapularis is basically a woodland tick. It requires that high humidity you find in the leaf litter and the forest floor or underground cover for survival, and that's where their primary hosts like rodents and ground nesting birds are. So if you look at 67 in the typical area, 67% of the population is out there in the woods. 22% are in the ecotone. Only 2% of the tick population is actually in your lawn area. So there are, much fewer ticks there. But even in the lawn, out of that 2%, 82% are within that 3 meters or 3 yards of the edge of your lawn with the stone walls or forested edge or ornamental standings. But again, 18% can be out a little further out the lawn. But again, we're talking about that smaller 2% of the population. So wooded edges, your stone walls, ornamental vegetation is where the majority, but not all the ticks are found. So the idea is to landscape a tick safer zone around the home, which is, where you're less likely to find ticks. And that's by putting in landscape barriers, spraying maybe just along the edge. And then think about your ground covers. I mean, I've collected ticks in Pachysandra right next to the garden hose, right next to the front doorsteps. So think shade, think cover, and think leaf litter, things like that. That's where you're going to find most of your ticks. So when you talk about your landscaping, here's a property that, had a lot of ticks. The number of ticks in the lawn area itself were reduced substantially by the way they managed or manicured the edge of the property. Stone walls, you can clean those up. They're essentially mouse hotels. A lot of ticks there. Leaf litter removal can reduce the number of ticks along that edge of the property by 49 to 70%. Landscape barriers, and of course in our experimental trials we used wood chips because that's what we could afford, but we reduced the number of ticks moving into the yard by 35 to 77%. And of course, think about your play sets, folks. Granted, where most of them I find when I'm visiting homes, because of the shade, they're back in the woods. That's in your tick zone. You're essentially asking your kids to go out and pick up ticks. So this would be a setup that would be less conducive to picking up ticks. Leaf litter management is an important factor. I've kind of alluded to the fact earlier that it impacts survival of ticks. And this graph will show you how that works. So here's the trials that were done here in Connecticut where we actually would remove or not remove snow and leaf litter. So here was no leaf removal and no snow removal. 94% of the ticks survived through the winter, the nymphal ticks. If we remove the leaf litter and as well as the snow, which is an insulator, survival was still high. It was 77%, but that difference was significant. And it was even greater in the case of Lone Star ticks. A recent study that came out from my colleagues in New Jersey, they looked at the impact of blown or raked accumulation of leaves at the long edge, and they found an increased number of ticks in those raked or blown accumulations, which basically brings up how you manage those, removing them off-site or bagging or composting. That leaf litter may reduce your risk as well. But you got to take that with a grain of salt because, you know, depending on where it is, leaf litter can be an important environment for many beneficial insects in your landscape as well. So when you're talking about landscape, a lot of people are concerned about exposure at schools and parks and campgrounds and things like that. And ironically, many parts of the school and park grounds may be already landscaped into what I call that tick safe zone, with lawns, mowed fields, playgrounds, and the buildings themselves. And it really depends on the amount of tree cover that you have. There's fewer ticks in the meadows or wildflower gardens. So that landscaping along the edge is extreme, like you see here, blowed mow grass here, you know, can be extremely important in reducing the exposure of people that are using the park area. And what about your trails? Okay, you obviously should still be taking tick bite prevention measures, but you know, how crowded are the trails? And obviously, I think last year and this year we saw an increase in trail and park use with those that were escaping quarantine fatigue. And even though we figure about the majority of Lyme cases occur around the home, that increased activity outdoors could obviously increase your risk of tick. So I call these your tick safer trails. They're wide. It's easy to avoid the vegetation along the edge, as opposed to something like this, where you're obviously going to put you at much higher risk of encountering a tick. Another aspect of landscape management is your invasive plants. There have been a number of studies that have found that tick numbers are much higher with exotic invasive forest understories than native forest understories. And so we found that there was, you know, abundance of adult black-legged ticks that were infected with Borrelia was greatest in areas of dense Japanese barberry, where it was, you know, very invasive. And similarly, a greater number of Lone Star ticks were infected with Ehrlichia was present in stands of invasive honeysuckle. And part of the reason is these dense stands provide an ideal microclimate for the ticks and great habitat for their rodent hosts as well. So the reduction and long-term management of barberry has been shown to significantly reduce the abundance of infected ticks. So if you got dense stands of barberry behind your house, I mean, that's just great tick habitat, and you may want to think about removing that, or in the case of honeysuckle, they found that removing the honeysuckle reduced deer activity in the numbers of Ehrlichia infected ticks as well. So that's another important thing to consider. Now, there's been a lot of effort looking at host-targeted tick control, primarily focused on mice, like the white-footed mouse, which is a reservoir for the Lyme disease spirochetes, and what I call the tick reproductive host, the white-tailed deer. Now, there's a number of approaches that target our rodent reservoir hosts, like the white-footed mice or eastern chipmunk. One are the so-called tick tubes. These are tubes that contain either Demonex or Thermacell, or the two products. They contain permethrin-treated cotton, which is taken by mice back to their nest and kills any ticks that may be present. Chipmunks don't use the cotton in the tick tubes, and there's been uneven performance across a number of field studies with these tick tubes. And of course, they would not apply to Lone Star ticks or Asian longhorn ticks, as the immature stages of those tick species don't use rodent hosts. Another alternative is the Fiprenel Bait boxes, and I was involved with the original studies with this. It contains 2 non-toxic bait blocks and a wick with Fiprenel. Fiprenel is the active ingredient in frontline for flea and tick control for dogs and cats. Very effective on mice. So the idea is the mice go in, and once they're treated, they'll be killing any ticks they encounter in the environment for up to 30 days. So you're basically killing the ticks as opposed to on the mice, rather than infecting new generation of ticks. But but you do have to bear in mind that a mouse home range or territory ranges from 0.2 to 5 acres, averages a little under an acre. And so you've got to really, if if you're using the tick tubes or the bait boxes, you have to space them around the perimeter of your property. 24 tick tubes will typically cover an acre, placed 10 yards apart twice a year. That's a total of 48 tubes. The number of boxes and pattern and placement for those Fiprenel bait boxes, they average about 10 per half to 1 acre size property, about a minimum of 30 feet apart. And so it will vary on your landscape. Tick tubes, basically, in one study, a recent study in New Jersey, they had 20% or so control of host-seeking nymphs year 1 and year 2 of their studies at a cost of about $3 a tube. And the Fiprenel bait boxes, though, provided almost 80 to 84% control by year 1 and year 2 at a cost of $45 a unit. So, but those are viable tools as part of your tick management program. There's also been a lot of focus on white-tailed deer, either by exclusion of the deer, reduction of deer numbers, or treatment of the deer. And fencing will reduce in one big study that I did, this is a 7-strand high tensile electric deer fence. Here's a typical regular type deer fence. Fencing reduced the number of larval, nymphal, and adult ticks by 100 to 74% inside the fence, because if the deer can't get in to drop female ticks to lay eggs, obviously you're not going to have any larval ticks, and you're going to have substantially fewer nymphs and adults as well. I'll get into the reduction of deer here in a moment, but another approach is treating the deer. This device is called a four-poster. It contains corn that the deer can then come up and feed out of these little troughs on either end. These paint rollers are treated with permethrin, and so it's a topical application device, and it kills the ticks on the deer. They get, treated when they come up to feed. So if we look at the impact of these deer-targeted methods on human Lyme disease, these are the few cases where we actually have some data showing an impact on human disease. The first, there's a large multi-state project that ran from 1997 to 2004, conducted in 5 states using these 4 posters. Here in Connecticut, it was in a neighborhood in Old Lyme. And a subsequent study looking at Lyme disease incidence there in Old Lyme versus some surrounding comparison control towns, we did find a significant reduction in Lyme disease cases and where we had the 4 posters. Looking at deer reduction, one good example is in Mumford Cove in Groton, Connecticut. Deer there in that community were reduced from about 122 deer per square mile, which is a very high density of deer, to 13 deer per square mile, over 87%. There was a 76% reduction in tick abundance and an 80% reduction in resident reported cases of Lyme disease. So this is a rough threshold. Densities of around 13 or 10 to 13 deer per square mile can significantly reduce the number of infected ticks, enough to reduce the risk of humans contracting Lyme disease. And in fact, going back to, remember I mentioned Manresa Island, where we discovered that really high population of Lone Star ticks? Well, it's actually a peninsula, and this is an area that's not open to the public. It is an area that has a decommissioned power plant. And so what we did is we put four poster devices on the site to apply the 10% permethrin, maintained through 2018, 2019, and 2020 through the summer months weekly. And then ticks were sampled over that same period. And we reduced the density of host-seeking Lone Star ticks by 93%, nymphs by 92%, and larvae by 96% by treating the deer to control the Lone Star ticks feeding on them. And we also documented a significant reduction in the number of ticks and the tick burdens on the white-tailed deer as well. As you can see here, when we started, the ears of the poor animals were just covered with ticks. And basically, when you're done, there just really aren't any. So how can we pull these tools together? There's been a lot of interest lately in integrated tick management, where we're using a combination of techniques, where you're talking about treating the deer, the mice, spraying, these things together. And so this is a summary of some integrated tick management projects that have been ongoing or just finished. And I have a review on this approach in the Journal of Integrated Pest Management. that was published in 2017. This journal is open access, so any of you that are interested in reading this review have ready access to it. So there's a couple of projects that we did here in Connecticut, and I'll kind of mention those here in a moment. There's also a study that was done in Fairfield County by Western Connecticut State University, and that's being written up for publication. And there was a much larger scale project done by the Cary Institute of Ecosystem Studies, funded by by a foundation and in part by CDC, again, using bait boxes and spraying the fungus similar to what we did here. And again, you can review this in that journal. But I'll just briefly mention one of our studies that was done in Reading, Connecticut, where we had 4 neighborhoods, where we had one neighborhood was a control. We actually tried deer removal in a couple of the other neighborhoods. And then we had the other neighborhoods were treated with the Met 52, which is that fungus spray I mentioned earlier, which is unfortunately no longer available, as well as that Fiprenel Bait box. And as you can see here, the number of ticks on the mice dropped dramatically where we had the combination of the bait boxes and spraying for ticks. So the combination of any of the treatments reduced questing nymphs at the home properties by 78 to 95% overall. And the encounter potential, what we running into a single infected nymphal tick on those properties was reduced by 66%. In 2017, we began another 5-year project, so we're wrapping that up here this year to evaluate the integrated use of the Fiprenel-based rodent bait boxes again. And instead of trying to reduce deer numbers, we actually use the deer 4-poster feeding stations, and again, applications of that entomopathogenic fungus, the Met 52 product, Metarhizium anasophyli, and various different combinations at the property, at the properties in 7 neighborhoods. And we're in the process of doing the final sampling here this year, as well as, and we'll be analyzing the results and so forth for publication here very shortly. Now, I do get a lot of questions on the Lyme disease vaccine. The original vaccine was called Limerick's, manufactured by SmithKline Beecham for use in 1998. And it's kind of unique because basically it was an outer surface protein A vaccine. So when you got vaccinated, you would produce antibodies to that outer surface protein A, which is what's expressed by the spirochete in the tick gut. So basically you're sterilizing the tick after it attaches to you and begins to take in that blood meal. So in clinical trials, it showed about 78% effectiveness after the required 3 doses. But some patients who got the shot went on to claim that they developed arthritis after the vaccination. The evidence for this was very weak, never substantiated. But given the low number of doses that they expected they would sell in 2002, they pulled the vaccine. Currently, there are 2 options in the works. One is a vaccine that works the same way that the Limerick's vaccine did, although they made some changes to it. It'll be more universally applicable to not only Lyme disease here in the US, but Lyme disease in Europe as well. And there's another is a 1-shot that would be taken annually in advance of the tick season. It's a pre-exposure prophylaxis. So Valneva, a French biotech company, and Pfizer has already completed their phase 2 trials, which are very promising, and they're going to begin phase 2 trials for children as well. And hopefully this, after they finish phase 3 trials, maybe we'll see a vaccine available by 2025. That would be terrific. And then Mass Biologics is developing this called PrEP shot for Lyme. This is a shot that would be immediately effective, unlike a advanced true vaccine, where you have to wait to build up antibodies. But it is through their phase 1 clinical trial, and they're hoping they can finish all those trials by 2024. So that'll kind of give you an update on where things stand on a Lyme disease vaccine. So where do we go from here? The growing number and spread of ticks and tick-borne diseases poses an increasing risk in the US. Ticks are widespread and difficult to control. We have a lot of multiple challenges or barriers to effective tick bite prevention and control. And these are very nicely summarized in a paper by Dr. Eisen and myself in the Journal of Medical Entomology published in 2020. This is open access, so you can readily get to it. We definitely need a lot more research on tick biology, ecology, the tick pathogens, the human-tick interactions, and tick integrated pest management approaches. As I kind of summarized today, we have a lot of tools available for killing ticks, but the impact on disease continues to be unclear or unproven. And a lot of these methods I discussed, there's not something that's really readily available or utilized by homeowners or on a scale. It'll be interesting to see what the role of a Lyme disease vaccine is coming in the future, but nonetheless, think about it. Any of these tick control tools, they've got to be effective. They've got to be safe. They have to be scalable, from a small location to a much larger scale, like a park or something. They have to be affordable. If they're too expensive, they're not going to be used. So they have to be actually used if they're going to be effective. So there are a lot of resources out there. I hope this talk has been helpful, but I want to point you, I do have a number of flyers available on our website, my tick management handbook, as well as some additional flyer informations. The CDC has a nice booklet on tick-borne diseases in the United States, as well as their website. The Northeast Regional Center for Excellence in Vector-Borne Diseases, of which the experiment station is a primary member, has a lot of resources for mosquito and ticks. And I would also point you to the National Institutes of Health Tick-Borne Disease Working Group. This was established by an act of Congress. And so their first report to Congress was in 2018. Their second report to Congress was in 2020. These were in 2-year cycles, and there will be another 2-year cycle for a report to Congress in 2022. These are available online. Very good overview of tick-borne disease in the United States, from diagnosis and treatment to prevention, and where things stand. Of course, all of the research that we do here at the experiment station couldn't be done without the efforts of what I call the CAES tick team, my collaborators here, technicians Heidi, Michael, and Jamie, and Duncan, as well as my postdoc, Dr. Megan Linsky and Dr. Eliza Little. Dr. Gudars Malay runs the tick testing laboratory. Dr. Scott Williams oversees many aspects of our tick control projects. He's a wildlife biologist. And Dr. Douglas Brackney oversees with Duncan the tick testing for the active tick surveillance program as well. So obviously it's a team effort for all the work that we do here, and I simply want to acknowledge their contributions. And I will finish up here with a couple of quotes. Pliny the Elder, the Roman writer, noted that ticks were the foulest and nastiest creatures that be. And an old prayer dating from somewhere around 1856, from red bugs and bed bugs, from sandflies and landflies, mosquitoes, gallinippers, and fleas, from hog ticks and dog ticks, from hen lice and men lice, we pray thee, good Lord, give us ease. Thank you.
Thank you very much, Dr. Stafford. That was an amazing overview of ticks, tick bites, and tick-borne diseases. We do have some questions in the chat, but I did want to give a shout out to Don Mitchell, who joined the talk, but also brought over very kindly a sample envelope, and I don't know if you could see this, a little tube for tick collection. If you happen to find a tick on you that's bitten you, you are welcome at free of cost. There's no charge to have a tick tested. They have these envelopes at the Essex Town Hall, and I'm going to assume that at most of the town halls in Connecticut, you can get them and have your tick tested just in case you're worried about having a disease born from a tick. So let's get right to the chat. And Alan wanted to know what is recommended for dogs to prevent the tick bites.
Okay, I didn't get into that. For your companion animals, there's a huge variety of products available either on the market or from your veterinarian that are quite effective. And so, and as well as there's several brands of Lyme disease vaccines that are available for dogs as well. And so the easiest way to do that, I would direct you to the Agricultural Experiment Station's website. And on there in our publications, I have a relatively new, nice little handout or fact sheet on controlling ticks on companion animals. And I list all of the products that are available within that fact sheet as well. So that would be an excellent way to answer your questions on that. And of course, anyone is free to contact me if they still have any questions after that.
Great, and I'll be sure to post links to some of these fact sheets and papers that have been written and posted and are publicly accessible. Alan also wanted to know, does mulch help or hurt?
Mulch can help, but bear in mind that you're looking for somewhat of a drier substrate. That forms kind of a barrier. If it's allowed to degrade, it won't be much different than the leaf litter. In the trials that I did, we actually laid it with black plastic, and then we kept brand new or fresh wood chips on top. Cedar chips can be quite effective, but they lose that effectiveness over about 6 months as the active materials in the cedar volatilizes and is gone. But it can be helpful. It also it's also a psychological barrier, so that this is your tick line. Step over it and you're out of the protected zone, so to speak. So it can have a dual function.
Very good. Thank you. Susan wants to know, why was Met 52 removed from the market, if you know?
That's a little unclear. It may be a sales issue. I haven't been able to find a lot of information on that. I suspect that the company is actually, I believe, has it for sale. And if they can't sell it to somebody else that wants to take over the Met 52 product, they'll just stop making it, which is really unfortunate because it's one of the few materials that we had as a biopesticide that was a true alternative to using chemicals.
Okay. I was curious, you may have mentioned it, but was Nucatone, is it safe for pollinators?
The Nucatone should be relatively safe for pollinators, but bear in mind that even in the essential oils, as I alluded in the one slide, that if it kills ticks, it can have some potential non-target effects. The Met 52 is one of the few things that had relatively few non-target effects, because it was basically a suspension of the spores. This fungus occurs naturally, but at very low levels. The background infection rate in the ticks is only 1 or 2% or so, not enough to provide control, obviously. So you're boosting the amount of spores out there and the chances of a tick encountering it. It had some effects on a few other tick species, but it was actually safe for our bees and many of our beneficial insects.
Okay. Well, if there are no other questions, I see a lot of thanks in the chat. Thank you so much, Dr. Stafford, for all this information and for allowing us to put the video up afterwards. So if you missed anything or you want to review anything that Dr. Stafford presented, it will be up later in the week, and we'll put it out on our social media that it's available. But keep checking our YouTube channel. It's the Essex Library Association. And you can see all of our other adult programs and kids programs on there too. Dr. Stafford, thank you so much for all this information, and thank you to you all for being in our audience today.
Thank you.
And stay well.
Okay. Bye-bye.
Bye.